Hi, my name is Dean Wingerchuk. I'm a professor of neurology and chair of the Department of Neurology at the Mayo Clinic in Phoenix and Scottsdale, Arizona. And today I'll be talking about approved therapies in NMOSD. These are my disclosures. The therapeutic strategy for attack or relapse prevention in NMOSD is expected to improve the natural history of the disease by targeting attack prevention because progressive NMOSD is quite rare. And this is in distinction from multiple sclerosis, where upwards of 2/3 of patients will develop a secondary progressive clinical course. We think that if we can prevent relapses in this disease, we can preserve neurologic function. The pathobiology of NMOSD is increasingly well understood, especially in patients who have the aquaporin 4 antibody. This antibody, which targets its antigen on the end feet of astrocytes, triggers a number of important immune modulators and effectors that we think drive the disease. In particular, complement is activated quickly after antigen antibody binding. B cells make antibody and have other effects on the T cell pathways that influence activity of the disease. And then interleukin 6 is upregulated by the antibody, stimulates plasmablast production of antibody, and also acts as a chemokine and a pro-inflammatory protein. The therapeutic strategies for attack prevention have, until recently, used unapproved immunosuppressive therapies such as rituximab, mycophenolate, azathioprine, or corticosteroids. But beginning in June of 2019, with the approval of the anti-complement monoclonal eculizumab, we now have a new era of treatment for patients with seropositive disease. We have 3 drugs. In addition to the complement inhibitor, we have the B cell inhibitor, the anti-CD19 inebilizumab, and the anti-IL-6 receptor drug, satralizumab. 4 randomized controlled trials have been completed for these drugs that are currently approved in the US. Eculizumab via the PREVENT study, the Nmomentum study for inebilizumab, and 2 studies, Sakura Sky and Sakura Star, for satralizumab. And I'll mention these studies as I go through and describe each of these drugs. Let's begin with eculizumab. This was the data from a phase 2 study of eculizumab in aquaporin 4 positive NMOSD. Patients had very active disease before screening. During treatment, virtually no attacks, and then after treatment, some of the attacks started to come back. This seemingly very favorable response led to the phase 3 PREVENT study published in the New England Journal of Medicine in 2019. The design was double-blind, placebo-controlled, and add-on design, 2-to-1 randomization with the primary outcome time to first adjudicated relapse. The interventions were eculizumab, 900 milligrams weekly intravenously for the first 4 weeks, and then 1,200 milligrams IV every 2 weeks thereafter. Placebo was matched in this 2-to-1 randomization. Who got into the trial? Patients who were seropositive and who had active disease, 2 attacks in the last year or 3 in the last 2 years, including 1 in the past year. EDSS of 7 or less, no mitoxantrone or rituximab recently, no IVIG within 3 weeks, and stable doses of immune suppressive therapies, commonly corticosteroids, azathioprine, mycophenolate, were allowed during the study. The doses couldn't be changed. Rituximab was not allowed. Prednisone was allowed up to 20 milligrams per day, and the dose could not be changed during the study. The PREVENT study was markedly and remarkably positive in demonstrating ability to prevent attacks. Overall, 43% of placebo patients and only 3% of eculizumab patients had attacks. This is illustrated in the top Kaplan-Meier curve. If you look at the middle and bottom curves, you see that the outcomes are similar whether patients were receiving or not receiving concomitant immunosuppressive therapy. So eculizumab seemed to have an effect regardless of whether it was used as monotherapy or as combination therapy. Secondary endpoints were evaluated. The adjudicated annualized relapse rate was also significantly impacted, but because of the hierarchical nature of the statistical analysis, other factors such as the EDSS, other measures of impairment, and quality of life were not evaluated. Adverse effects, there was one death in the trial, a patient who developed a pulmonary empyema, thought unlikely to be related to the eculizumab drug. Otherwise, the drug was actually very well tolerated. Upper respiratory tract infections and headaches were the most common side effects. There were no patients with Neisseria meningitidis during this trial. And of course, that is one of the main risks of eculizumab. Inebilizumab is the second treatment that we'll discuss. This is an anti-CD19 therapy. It's a monoclonal antibody that depletes B cells and has a somewhat wider range of B cell lineage depletion than does rituximab. You can see here that the anti-CD20 rituximab depletes early immature B cells all the way up to memory B cells, but does not impact plasma cells and plasmablasts. But the anti-CD19 inebilizumab does in fact have an effect on those cells. The Nmosd study was a double-blind randomized placebo-controlled trial with a 3-to-1 randomization and pure placebo. That is, patients were not allowed to be on an immunosuppressive therapy of any other type during the trial. They had to have active disease, 1 attack in 12 months or 2 in 24 months, and they did include some seronegative patients, but it turned out that the overwhelming majority were seropositive for the aquaportin 4 antibody. The drug is given 300 milligrams IV at days 1 and 15, and then 600 milligrams IV every 26 weeks thereafter in the study. So essentially dosed with a frequency similar to that of rituximab. Prednisone could be continued during the study for days 1 to 14 at an equivalent of 20 milligrams per day, and then tapered by day 21. This graph shows the circulating CD20 B cells in the trial, and as expected, the inebilizumab arm basically went to 0 within 4 weeks of the onset of treatment. Overall, the study was also positive with at about 73% reduction in the risk of first relapse or time to an adjudicated attack. And you can see the intention to treat population on the top and the aquaportin 4 positive patients only on the bottom. The results of the trial were driven almost entirely by that seropositive group. Out of 230 subjects, only 17 were seronegative. This study was also able to show during its 6-month randomized controlled period, was also able to show that disability was lessened compared to placebo as measured by EDSS. The number of hospitalizations was reduced, and overall MRI lesion burden was reduced as well. Of the 17 seronegative patients, 13 were randomized to inebilizumab, and 3 of those patients had attacks. Because of the low numbers and the distribution of randomization, there's little that can be interpreted from these data. Safety of inebilizumab was quite good. Overall, the types and frequencies of adverse events were similar between the drug and placebo, including infections and infusion reactions. There were no malignancies. There were 2 deaths in the open label phase. One was respiratory insufficiency related to an NMOSD attack, and another was an indeterminate neurologic death in which a variety of etiologies were considered. Finally, treatment targeting interleukin-6 and its receptor is important. Some early observational studies suggested that using the monoclonal antibody tocilizumab could reduce activity of the disease even in people that had very active seropositive disease despite rituximab. So again, there were two studies supporting the approval and use of satralizumab in NMOSD. The first was called Sakura Sky. Sakura Sky was a double-blind study with 1-to-1 randomization and the familiar primary outcome of time to first adjudicated relapse. To get in this study, subjects had to be 12 to 74 years old, so there were somewhat younger patients, even in the pediatric age group in this study. They could be seropositive or seronegative, and they had to have 2 attacks in 2 years, one of which occurred in the last 1 year. The interventions were satralizumab given 120 milligrams subcutaneously at weeks 0, 2, and 4, and then every 4 weeks thereafter. In this particular study, Sakura Sky, it was an add-on approach. That is, patients could continue azathioprine, mycophenolate, or corticosteroids. The results of the trial were positive. In the overall population, a 62% reduction in risk, that is time to first adjudicated attack. In the seropositive group only, this went up to 79%. This was the results for the seronegative group, suggesting that the drug does not have a favorable benefit here. Adverse events, no unexpected adverse events, no deaths, no neoplasms. Rates of serious infection were similar between the drug and placebo. The second study, the Sakura Star study, was a very similar design. The differences were that it was adult patients only, and that the interventions were satralizumab at the prior dose and frequency versus placebo as monotherapy. So here, patients could not be on an immunosuppressive therapy of another type. The results here were very similar to the Sakura Sky study. The protocol-defined relapses overall were significantly reduced, but when you stratified it by serostatus, as shown here, in seropositive patients, the risk of relapse was reduced by 74%, but there was no significant effect in the smaller group of seronegative patients. Here again, there were no unexpected adverse effects or deaths. So in summary, there's strongly positive results for these drugs in aquaportin 4 positive NMOSD patients. These trials confirm the pathobiologic relevance of complement, B-cell mechanisms, and interleukin-6 in this disease. The benefits are seen in the aquaportin 4 positive group. Overall, we see that this autoimmune astrocytopathy can be influenced by eculizumab, inebilizumab, or satralizumab, all of which provide exciting opportunities for patients to be able to intervene in this devastating disease as early as possible and to protect their function. Thank you very much for your attention.